Okay, the topic for today is drug discovery, development, and also design. This is under Module 3, and as you compare this module from the previous modules, mas maikli siya and easier to understand. Now, in drug discovery and design, you have to realize that it keeps on changing. So nagbabago siya from time to time. So the ones that I will be discussing right now is the current trends. Okay? Ito yung currently applied. And also, please drink this in that the practice nowadays was is a combination of all the learnings that the scientists obtained or acquired from the previous experiences. Siyempre, from the previous experiences, may mga good and bad learnings silang na-acquire na pwede nilang i-apply or iwasan. Kaya lahat ng i-discuss ko ngayon based from the learnings that they had from the previous practices that they had. Okay? So para magkaroon kayo ng idea kung ano yung nangyari dati or ano yung practice dati, let me give you some overview or brief historical background about the practice before. So dati, ancient people use potions, herbal medicines in crude forms. So when we talk of crude forms, they are not yet purified. Ito lang yung mga dinurog na mga halaman, dinurog na mga natural sources, okay, na ginagamit for certain diseases. However, because of that practice, it was discovered na mataas ang risk ng toxicity, ay mataas ang risk ng mga unwanted effects. That's why in the 19th century, ang naging goal before is to extract the pure substances from the crude sources. So kumbaga, yung raw source or yung raw plant for instance, hinahanap nila, kinukuha nila yung mismong active compound there. Okay? And because of that practice, they tend to realize that some of the compounds are desirable and some were not. Okay? Ganun yung nangyari. So in other words, before, hindi puro, and then naisip nila na hanapin kung ano ba talaga yung mapapakinabangan. Okay? And in relation to that, they were able to come up in the discovery of the so-called leads, okay, or lead compounds. So this is being pronounced as lead. This is different from the element lead. Okay? And later, we will focus more on the lead compounds, kung ano bang ibig sabihin niyan. Familiar na siguro kayo sa term na lead compounds, di ba? Sa dosage form ito na encounter, am I right? Tama, no? Dosage form ang last semester nyo? Familiar or no? It's okay if newly heard siya. So maiintindihan naman natin siya for this meeting. Okay? So lead compounds. Mamaya maiintindihan nyo siya. So yun yung dating nangyari. Okay? So in connection to that, let's now proceed on the current trends or yung present na practice in drug discovery, design, and also development. This is being categorized into three major steps. So steps ito hindi siya simultaneous na nangyayari. Okay? So number one is to find the lead. Second, we have to design the drug. And then finally, to develop it. So ngayon, unahin natin ang finding the lead. So i-define na natin ngayon, although naka-state ito sa isang slide later, para may idea na kayo kung ano yung magiging flow of our lecture regarding finding the lead. When we say lead, okay, lead or lead compound is the compound, compound with a significant pharmacologic activity. So this is a compound or chemical compound coming from a particular source with a significant effect or pharmacological activity in the body. When we say significant pharmacologic effect, meron siyang value clinically sa atin. Okay? That clinical value, of course, must be desirable. Okay? It must be desirable. Pag sinabing desirable, therapeutic. However, you must realize also that in reality, it's not always the case. Hindi sa first or hindi literally, pag maghahanap ka ng lead, 100% perfect na siya, 100% desirable na siya. So mamaya magigets nyo kung anong ibig kong sabihin dun sa reality na sinasabi natin. Okay? So under finding the lead, we have different steps to be followed. Number one is to choose the disease, followed by choosing the drug target, and then identifying the biological assay to conduct. Next is to finally find the lead compound. Next, we have isolation and purification, and if necessary also, structure elucidation. So ito yung mga steps to finally, okay, get the lead compound from a particular source. Following so far? Okay, so start tayo. Choose a disease. Guys, when you find the lead compound, if you want to discover a lead compound that has a significant pharmacologic activity, hindi siya shortcut na maghanap ka. Napakalaki ng mundo para maghanap ka ng gamot na may effect agad. Okay? Or ng compound na may desirable effect. Paano mo malalaman yon? Siyempre, meron kang mga ipra-prioritize na steps. Number one is to choose the disease. Guys, in reality, business pa rin ang pharmaceutical firms, and yung drug discovery, it's purely business. So what I mean to say here is second line or second in line na lang or next in line na lang yung inyong commitment in finding the cure, sa totoo lang. Kasi pera pa din talaga. So market first, then science follows. Ibig sabihin, pharmaceutical firms and research institutions, for example, they search for trending diseases, let's say, na pwede nilang i-cater, pwede nilang solusyonan. So that's why a huge investment should be made by research institutions and pharmaceutical firms in searching for new drugs and develop them. Okay? Major considerations are, number one, economic. So pag sinabi economic, kaya ba ng pharmaceutical firm na mag-invest o mag-spend ng napakalaking pera to be able to find a drug? And then medical, kay medical consideration. So I'll give you an example. Siyempre, very timely ngayon yung COVID-19. So because of COVID-19, nagdi-discover or nakapag-discover ng COVID vaccine. Do you think magkakaroon ng urge or magkakaroon ng inspiration yung mga pharmaceutical firm to spend so much to discover COVID vaccines if not for that disease? Syempre hindi, di ba? What else? For instance, economic and medical considerations. There are diseases that are particular in certain countries. For instance, tropical countries. Dengue, di ba? Dengue. Because of dengue that is common in tropical countries like in our country, Philippines, nagkaroon ng inspiration ang ibang companies to discover or to find vaccines like yung Dengvaxia and also mga anti-malarial agents because endemic sa atin ang malaria. Something like that. Okay? So in other words, before you find the lead compound, you have to choose a disease. So just like in research, when you have your research in the future, before you test some compounds, test some plants, for instance, or microorganisms, you have to know the problem. You have to know your chosen disease. So yun yung unang step. Can you follow? Okay, next. What about the second step? Under pa rin ng finding the lead compound is, second step is to choose a drug target. Ito medyo familiar na kayo because we were able to discuss Module 2 drug targets. When we say targets, these are macromolecules that are targeted by drugs, and those agents will modify the function of those targets. Okay? Take note that you have to understand the molecular biology of those macromolecular compounds for you to be able to establish your goal. Okay? That is, hanapin mo kung ano yung pwede mong targetin na related sa chosen disease mo. Okay? So in connection to that, let me give you an instance. Okay? I-combine natin, let's combine the first and the second step. Again, what's the first step? Is it target or disease identification? First step, disease, very good. And then, in connection to that chosen disease, you have to select your intended target. Okay? So, bigay tayong example. Halimbawa, in the Philippines, very common ang sakit na hypertension. That's why numerous pharmaceutical companies, they eye hypertension as their chosen disease. And you will learn later on, especially in pharmacology class, that we have different proteins or macromolecules that can contribute to hypertension if ever there is an abnormality already. Okay? So ano yung mga examples ng proteins na pwedeng ma-connect natin sa hypertension if ever those proteins are abnormally functioning already? Okay? Number one, pasok dito ang mga receptors. There are responsible receptors that could lead to increase in blood pressure. Okay? For example, these are just representative examples, okay, but not limited to them. So examples, we have angiotensin receptors, okay, angiotensin, okay, receptors. And aside from angiotensin receptors, kasali rin dito ang vasopressin receptors. These are, again, just representative examples. Okay? So kasali dyan ang mga receptors. And also part ng pag-contribute sa increase in blood pressure is because of different co-transporters, which are membrane proteins that allow exchange of compounds. Okay? Ito yung mga tinatawag nating transport proteins kahapon. Naalala nyo ba yung mga receptors and transport proteins kahapon? Okay, very good. Because abnormality of these co-transporters could lead to increased blood volume. Okay? Increased in blood volume. Okay? So co-transporters that could lead to increased blood volumes. Kasali rin for instance ang mga ion channels. Okay? Included din ang mga ion channels that could lead to constriction of blood vessels or vasoconstriction. Again, we can also categorize ion channels as proteins that could contribute to increased blood pressure because of vasoconstriction. Okay? So ito yung ibig sabihin natin ng mga contributory proteins that could participate to increase BP resulting in hypertension. So taken all together, ano yung ating takeaway sa dinidiscuss right now? The point here is if a pharmaceutical company eyes hypertension as their target disease, they can select their target proteins na pwede nilang i-test using a certain compound of interest. Okay? Kaya you have to know the physiology of these proteins kasi related yon sa inaay nyong sakit. Nagets ang connection ng 1 at 2? Okay. Next, punta na tayo sa number 3. After we realize that we have to choose first the disease and also the target protein, syempre isipin nyo if you were able to identify your drug target or a particular protein in the body, you cannot get it easily from a person. Hindi mo naman pwedeng himayin yung receptor, kunin yung mga proteins, mahirap yon. That's why, in other words, you need an improvisation or improvised, right? Parang ganyan. So in connection to that, you have to identify a biological assay or testing method. Okay, testing method. So sa testing method, we have two types of testing methods. So these are in vitro and in vivo testings. So later we are going to differentiate in vitro from in vivo tests. But in reality or usually, ang ginagawa ng mga pharmaceutical firms, they start with in vitro first and then in vivo. So ano ba yung difference in general ng in vitro at in vivo? Ito muna yung sasabihin natin kapag in vitro, usually walang living organisms. Okay? Walang ginagamit na buhay initially. Okay? Kapag in vitro testing. Pag in vivo, gumagamit na dito ng buhay like animals and even humans. Okay? Like animals and in some cases humans. So experimental animals and humans. In other words, merong life na involved na. Okay? In practice, ito yung ethical way of testing. In vitro first and then in vivo. Why do you think so? Bakit kailangan mauna ang in vitro bago ang in vivo? Why do you think so? Any idea? Sige, okay lang. Ano sa tingin nyo? Bakit kailangan unahin ang mga instruments bago ang in vivo test? Bakit? Sa tingin nyo? Sige, kahit isa lang sumagot, kung ano lang, pwede mag-unmute. Yes, very good. Para walang masayang na life. Risky. Sige, tama yun lahat. I-take natin yung mga answers sila. Tama. Ganito yon. Isipin nyo ha, pag kayo ay magte-test ng isang compound, pag kayo ay magdi-discover ng gamot, tanong ko sa inyo, pag mag-experiment pa kayo, 100% positive yung result, yes or no? No, hindi. Paano pag inuna mo yung tao tapos namatay siya agad o yung animals namatay, sayang yung buhay. Okay, sayang yung buhay. At least pag nag-in vitro ka, kung mag-negative man yung result mo, hindi siya unethical. Okay? Kumbaga, makatao pa din yung nangyari. Okay? However, ito yung nangyayari sa Pilipinas lang ha, sa Pilipinas. Hindi ko sure sa iba. Okay? Sa Pilipinas, hindi ito nasusunod kasi sa totoo lang, ang in vitro test sobrang mahal. Mas mahal pa to kaysa sa daga. Kaya usually ang nauuna sa atin, animals bago mag-in vitro. Kasi in the interest of money, inuuna yung animals. Kaya mapapansin nyo sa mga thesis, sa totoo lang kung nakarinig na kayo ng mga defense, ganyan, 'di ba pansin nyo minsan, animals agad ang tinetest sila, tinest agad sa daga. Sa totoo lang, hindi siya scientifically sound. Hindi siya ethical, parang ganon. So dapat in vitro muna. Kaya lang yun nga sobrang mahal. Para may idea kayo sa sinasabi ko mahal, na ganito ako, inuna ko naman yung in vitro bago in vivo. Yung in vitro test, 60,000. 60,000 yon, isang kit lang. Isang ano lang, parang basta kit ang tawag doon. And then sa in vivo, siguro gagastos ka lang na around, mahal na yung 20K. Marami na yon. So napakalaking difference talaga. So yon, pera. Pera talaga ang magkocontrol. So yun yung difference. So para magkaroon kayo ng idea further between in vitro and in vivo tests, let's differentiate them. First, we have in vitro tests. As I've said, no life is involved. Usually ginagamit dito specific tissues, cells, or enzymes. Sir, buhay din naman yung tissues yung cells or yung enzymes oo, buhay sila pero wala na sila sa tao, they are being cultured already in the laboratory. So in short, they are cultured, they are being placed in a certain container. So wala na sila sa buhay. So kung masayang man to, hindi buong buhay ng tao or ng animals ang nasasayang if ever negative. Okay? So that's in vitro test. As long as there's no life being compromised or involved, in vitro test. Now, in case that the test becomes positive in vitro, you all have the guts now or confidence to proceed to in vivo testing. In vivo testing, you need animals or in some cases nga humans, and this involves testing clinical, okay? Clinical testing wherein you have to observe the symptoms if ever meron na nagmanifest sa patient. Okay? Question, kung sayang sa buhay ang in vivo, bakit kailangan pa rin siyang gawin? Bakit kailangan pa rin siyang gawin kung delikado sa buhay ng tao? Sa tingin nyo ba, mas malaki ang impact pag hindi nag-in vivo tapos minarket yung gamot? Mas malaki ba yung risk? Yes, mas malaki yung risk nun because the purpose of in vivo test is for you to simulate the effect of the drug inside the body of a living organism. So the point here is you have to identify if the kinetic profile of the drug is okay and also dynamic profile of the drug is good inside the body. Kasi chances are, take note of this, this concept. Chances are, for instance, you conducted in vitro test and the test became positive. Maganda result. Pag magandang result sa in vitro, puro kasi ang in vitro cells na mismo, diretso na dun Pero syempre, pag binigay mo sa tao, do you think pag binigay sa tao, maraming possible obstacles ang gamot? Yes. So the point is, pwedeng positive sa in vivo, sira na yung gamot kasi ang daming mga harang or hindrances inside our body that could prevent the drug from reaching its target. Kaya in vivo test has to be conducted as well. Okay? So yun yung mga tests na kailangan gawin. So tapos na tayo ngayon sa identification of bioassay. Okay? Next, number 4. So natapos natin ang choose the disease. Second is to identify drug target, and then bioassay. Now, after we discuss the first 3 steps, we can now find a lead compound. Ulitin natin, lead compound is a compound that shows a significant pharmacological activity or desired pharmacological activity. So this is how it goes. In finding the lead, for the second and the third step, lalong-lalo na sa third step, we have numerous compounds being tested. For example, you have 100 compounds that you tested in vitro. So out of 100, hindi lahat yon ay magpa-positive in vitro, iilan lang. Lahat na magpa-positive doon, lead compounds ang tawag sa kanila. Meron silang significant activity that you could further test. Okay? That's why it says here, the lead compound provides a start for the drug design and development process. Additionally, when you find a lead compound, pag nakakita ka ng lead compound, it does not 100% mean that they are safe. Yes, they have activity, pharmacological activity. They could be therapeutic, however, they could be toxic as well. Kaya ano pa lang to, candidates. You really need to design pa, you really need to develop pa. Pero at least, meron kang starting point. Okay? Kasi napatunayan mo, meron siyang initial activity. So that is the lead compound. Gets? Nagigets ba? Okay, so let's proceed to the different ways in discovering a lead compound. Guys, meron tayong iba't ibang paraan to be able to find a lead compound. So we have the most common one. So inilagay natin dito most common. Okay? First, we have natural products screening. Second, we have medical folklore. Next, we have screening synthetic compound libraries. Next, there are cases wherein existing drugs serve as leads. Next, computer-aided design. And finally, serendipity. So iisa-isahin natin sila. Number one, the most common and ang pinakamatandang way to find a lead compound is the natural products screening. When we say natural products screening, lahat ng kayang i-provide ng environment, we screen them. Okay? So most biologically active natural products, the potential lead compounds are usually secondary metabolites. So when we talk of secondary metabolites, these are the products formed when the biological molecules like carbohydrates, like proteins, like lipids were metabolized. Okay? So secondary metabolites yan. You will learn secondary metabolites in pharmacognosy. Okay? Pharmacognosy. That's why related subject or field here is pharmacognosy. Now, what are the examples of natural products? So when we say natural products, as I've said, these are being given by the environment that could serve as sources of leads that we can develop as drugs. So number one, pinaka-common plants. Kaya pansin nyo sa mga thesis na nakikita nyo, usually ang tinetest nila halaman, di ba? Kasi natural product ang halaman. Pero meron pang iba. Halimbawa, we have microorganisms, marine life, animals, and even venoms and toxins. Sa plants, sobrang daming examples dito. So I will no longer cite examples sa plants kasi you will have pharmacognosy later. Okay? Sa microorganisms, ang dami din. We will be able to discuss this when we proceed to final grading period. Kasi kasali dyan ang mga antibiotics kasi they are of microbial origin, antifungal agents, and antiviral, anti-neoplastic. These are coming from microorganisms. Okay? Kaya mas maiintindihan nyo to pagdating natin sa mga latter portions. Marine life, yes. Yung mga algae, okay? Nagiging source na sila ng therapeutically active compounds. Animals, yes. And also we have venoms and toxins. So ang daming pwedeng ibigay ng environment to search, to be able to search for drugs. Kaya ito yung pinaka-conventional, pinakamatanda at pinaka-common to everybody. Okay? Pero take note, we must not limit ourselves sa natural products screening because we also have other ways to discover lead compounds. Pwede ring medical folklore, traditional folkloric claims. Anong idea natin sa medical folklore? Basically, pag medical or traditional folklore, ay familiar ba kayo na may mga albularyo, may mga sabi-sabi yung mga kapitbahay, uy, yung halaman na to, pwede ito sa ganitong sakit. Uy, sabi nila, pwede raw ito. Sabi-sabi, chismis. Now, Ang chismis ba may ebidensya? Usually wala, di ba? Wala. So dito, okay, sa medical folklore, somehow connected siya sa natural product screening because this will serve as your inspiration to test that compound, to test that plant, to test that extract, for instance. Kasi ang dami nagsasabi in the community that that drug or that herbal compound is useful for hypertension, for inflammation, for pain, and so on and so forth. So ang point is, you will verify if that is true or false. Siyempre, 50-50 pa rin yun kasi sabi-sabi walang ebidensya, so it could have a good result or negative result. Okay? Medical folklore ang tawag doon. Next, screening synthetic compound libraries. So what do we mean by that? Pharmaceutical companies also invest in their compound libraries. When we say compound libraries, meron silang computers, they have systems wherein they just save their compounds there pero not yet tested. Okay, not yet tested. Nandun lang, nakasave lang. Now, paano yun nadadagdagan from time to time? Nadadagdagan siya from time to time because they often collaborate with universities, with research institution na nakapag-identify ng isang structure o compound sa isang natural product o kaya sa isang source. So ibig sabihin nun, they were able to identify the compound pero they can no longer test it because of financial reasons. Kaya ang gagawin nila, ibibigay nila sa mga pharmaceutical companies. So ang gagawin ng pharmaceutical companies, iipunin lang nila yon. Iipunin, nasa computer lang. Tapos kung anong trip nilang kunin, o sige, test natin to. Kunin natin sa library. Kunin, test. Random lang. O sige, kunin natin to. O test natin to. Tapos boom, biglang may lead compound na. Okay? Kaya sugal talaga yung pag-discover ng lead. Sugal siya. Nagets niyo ba yung library? Parang ano siya, parang para siyang raffle. Parang, o sige, try natin to. Try natin to. Trip, parang ganon. Siyempre, depende rin sa sinasabi ng mga kakolaborate nila. Factor din yon. Gets ba? So more of ano lang to, kwentuhan yung drug discovery. Okay? Nothing too serious. Pero sa mga task at saka sa exam, syempre application to. Okay, application. So when you study this, hindi ako mahilig sa naka-base sa PowerPoint dito. Halimbawa ito, naka-base sa PowerPoint. Application to. So pwede ako mag-cite ng instance, ng case, tapos isipin nyo, ano kayang klaseng discovery ang nangyari dito? O, binibigyan ko kayo ng idea kung paano ako magbigay ng assessment. Okay? So kung nag-gets nyo yung principle sa bawat portion ng dinidiscuss ko, medyo may isip nyo, anong applicable sa case halimbawa na ipepresent natin, either in the exam or in the task. I hope you got my point. Nakuha? Okay. Next. Ito, existing drugs. Ito exciting to kasi ito yung mga parang mga war na mga networks, ay mga network wars in GMA versus ABS-CBN. Di ba pansin niyo sa mga networks, nagkokopyahan lang naman sila ng mga shows. O meron din ganon sa pharmaceutical companies. So meron tayong isang paraan para malaman ang lead compound, ang tawag doon existing drugs. So under existing drugs, we have two. Okay, we have two. Number one, me too and me better drugs and also enhancing a side effect. Ayun yung dalawa. So unahin natin ang me too and me better drugs. Ganito yung style sa me too and me better drugs. One pharmaceutical company, a pharmaceutical company was able to discover a drug. Now, that drug na unang na-discover ng isang pharmaceutical firm, kokopyahin ng iba, yun mismo ang imamodify nila. Okay? Ganun ang gagawin. So yung lead compound nung ibang pharmaceutical company, yung na-discover nung unang pharmaceutical company. Nagets nyo? Nagets nyo? Parang aanuhin lang sila, kokopyahin lang. Actually, hindi siya kopya sa dulo kasi minodify yung structure. Pero syempre kung bitter kang ikaw una nakadiscover, parang, naku, kinopya lang yan sa amin yan. Parang ganyan. Okay? So many companies use established drugs from their competitors as lead compounds. So the aim is to modify the currently available drug from the first pharmaceutical company so as to avoid patent problems and to be able to improve the profile of the drug. Okay? So let me give you an example. No need to copy the structure. I won't, wag nyo na copy yung structure, pero at least yung mga pangalan ng gamot ma-identify nyo. Okay? So for instance, we have captopril. A pharmaceutical company, although hindi ko nalagay, pasensya na, at hindi ko rin na-search kung anong pharmaceutical company ang una nakadiscover ng captopril. So pwede nyo i-research yun sa internet, okay lang naman. So ang isang pharmaceutical company was able to discover captopril. Now the problem with captopril is that meron siyang mga incompatibilities in the GI tract. Kaya this served as the lead compound for other pharmaceutical companies like Hoffman, La Roche, and Merck, and also yung Merck pang isa. So para maka-improve sila sa captopril, kinuha nila yon as lead compound and then they modified the structure. Kaya meron tayo mga iba pang ACE inhibitors like Cilazapril by Roche, and then Merck was able to produce two related compounds to captopril like lisinopril and enalapril. Okay? So these agents are said to be better than captopril. Now, may hit ba sa unang pharmaceutical firm? Wala, kasi bagong compound sa to eh. Nakuha nyo? Ayun. Gaya-gaya, parang sa mga phones lang, sa mga cellphone, di ba? Wala namang ano. So, uso rin yun sa pharmaceutical firm. Pera-pera lang talaga yan, guys. Okay? Ma-realize yan lalo pag tumatanda kayo. Pera-pera lang yung ibang aspeto. Yun. Next, continue natin. Pwede na sa enhancing side effect. Okay, so next, proceed tayo sa enhancing side effect. What do we mean by enhancing side effect? There are cases wherein the side effect of a drug could serve as the company's opportunity to make it a therapeutic application of the drug. So ulitin natin, there are cases wherein a given side effect of a drug could serve as its clinical use. Pwede siya maging opportunity to earn money, parang ganon, okay? By modifying the structure of the drug. So let me give you a scenario. So dito medyo confusing yung nakalagay, kaya bigyan ko kayo ng flow. Okay? Ito yung flow na nangyari dun sa previous slide. Do you have this? Itong naka-present right now. Do you have this one? Pero ito wala. Wala nito, di ba? Pero yan din yon, okay? Yan din yung nasa previous slide. So makinig sa akin. Pwede niyong picture na lang mamaya after kung ikwento ito. Okay? So the first or the story behind this was medicinal chemists during that time were discovering agents that would act in the autonomic nervous system. So they were searching for an autonomic drug. But upon discovering benzodiazepines, ito yung na-discover nila, that could act in the ANS, they were able to identify also that benzodiazepines can block histamine 1 receptors. Okay? So they can block histamine 1 receptors. So naisip nila, benzodiazepines can block H1 receptors. So this could serve as an opportunity for benzodiazepines to have another clinical use. Okay? That is anti-allergy. Okay? Pwede siya maging anti-allergy. Kaya they improved the structure of benzodiazepines up until they were able to extract ethanolamines, chemical compounds ethanolamines, in which ang example dito is diphenhydramine. You know diphenhydramine? Okay, so you are familiar with diphen. Ang diphen pala is galing sa benzodiazepine. Okay? It came from benzodiazepine. Diphenhydramine is a well-known anti-allergy medication because it blocks H1 receptors. Okay? And further modification of ethanolamines, medicinal chemists were able to get ethylene diamines chemical structure, where triphenyllamine is an example. Just like diphenhydramine, it is an anti-allergy med. Okay? Common anti-allergy med siya. So anti-allergy, anti-allergy. So what happened next? Okay? Ano nangyari nung sumunod? Nawili ang mga medicinal chemists. They further modified the structure of triphenyllamine and diphenhydramine. So while improving the structure or while modifying the structure of diphenhydramine, they were able to discover that because of the anti-allergy effect of these compounds, nagkokos ito ng sedation. Okay? Nagkokos sila ng sedation. So these agents, diphen and triphenyllamine, can cause sedation. Now, at a first glance, you can think that sedation is a bad thing. But medicinal chemists, maparaan sila. Naisip nila, pwede natin itong gawing clinical use later on. So what they did, they modified ulit the structure of these antihistamines until they extracted promethazine. Nakuha nila yung promethazine. Promethazine is a drug that can cause extensive sedation. That's why ang clinical use nito, anesthetic drug, okay, anesthetic agent. Meron tayong pharmacological agent na promethazine na available sa market. Okay? So ang use niya pang anesthesia. So anong napupulot na aral dito? Kaya nagkaroon ng promethazine na anesthetic agent kasi nakitaan yung pinanggalingan niya ng side effect na sedation. Nakuha? Nagigets nyo? Okay. And finally, pinaglaruan pa rin nila, they further modify the structure of promethazine until they discovered chlorpromazine. What's with chlorpromazine? Chlorpromazine has a tranquilizing effect. Okay? So tranquilizer siya, tranquilizer. So pag sinabing tranquilizer, pampakalma. Okay? Pampakalma, that's why ang clinical application, the clinical application of chlorpromazine is antipsychotic agent siya. So ano ang takeaway natin overall in this given figure? Side effect based siya. Nagkaroon tayo ng tranquilizer because of the anesthetic effect of promethazine. Nagkaroon tayo ng anesthetic agent promethazine because of the sedative effect of the antihistamines discovered. Okay? Paano natin nalaman na may antihistamine effect ang mga ito? That's because scientists discovered that the former drug benzodiazepines, meron silang H1 blocking effect. So that is based on side effect. Did you get it? Huh? Okay, so that's based on side effect. Napakadaming gamot din available in the market na based sa side effect. So it's for you to find out sa mga susunod na mga madidiscover o mapapakinggan yung knowledge. O kaya sa mga task or exam, baka may mga case na ganito, at least ma-picture out nyo kung ano nangyari. At least alam nyo ang scenario o yung principle niya. Okay? Pwede na po sa computer-aided design. Okay, moving on, we have computer-aided design. In computer-aided design, this is quite advanced because molecular modeling software programs are being used. So in this case, medicinal chemists, pharmaceutical companies use computer systems in identifying the binding site, in studying the binding site of the target, and also identifying the binding groups from the drug or the lead compound. Diba guys, remember kahapon, I was able to show you a hypothetical example of binding between a drug and its target. Are forces of attraction essential there? Okay, so recall tayo, what are those intermolecular bonds? Pag between positive and negative, what is it? Very good. Okay, ionic. Pag between nonpolar compounds, what is it? Yes, van der Waals. And then, pag hydrogen bond, saan nagpo-form ng bond ang hydrogen atom? Come again? Saan? Hydrogen bond, yung hydrogen atom, saan siya nagpo-form ng bond? Sa anong tawag doon? Electronegative atom. Okay. Very good. So, ang ginagawa dito sa computer-aided design, yung binding. Okay, yung binding. Of course, you will be able to modify the effect of the drug or enhance at least the effect of the drug by altering the binding ability of the drug to its target. So yun yung computer-aided design. And then finally, ito ay isang way din to searching for a lead. Serendipity. Familiar ba sa term na serendipity? Yes or no? Before I explain, just to check. Very good. Yes. Ito yung parang, may na-discover ako, parang ganon. Di ba may mga bagay na hindi mo pinagplanuhan, pero nangyari siya. Okay, nangyari. Wala siya sa timeline mo, wala siya sa mga standard operating procedure na plano, pero may na-discover kang significant. So God's God's plan. Frequently, lead compounds are found as a result of serendipity, by chance. Example, penicillin, di ba? Madidiscuss natin, we will be discussing penicillin later on. So I will state the story, kukwento ko sa inyo later on kung ano yung nangyari dun kay nakalimutan ko yung nakadiscover, si Alexander Fleming nga ba? Nakalimutan ko, sorry. Tama. So yun, si Alexander Fleming. So kukwento natin later on bakit siya serendipity. By chance. Ay walang plano. Pwede yon. Okay? So those are the different ways on how to find a lead compound. Overall, 19 yung 7 ways. Ay 19 yung 7 ba yun? I guess, 6. Ay 6 ways to discover. Okay? So na-discuss na natin sila lahat. So going back, we have natural products, medical folklore, compound libraries, existing drugs, computer-aided design, and finally we have serendipity. Moving on, guys, we were able to discuss finding the lead compound. Remember, once we found the lead compound, it's not yet 100% good or desirable. Of course, we need to further purify it and isolate it to be able to design the rightful structure for that lead compound. So if needed, we can perform isolation and purification methods, but this is somehow tedious. Mahirap siya. If the lead compound is present in a mixture, you need to isolate that to prevent interaction with other compounds. And finally, we have structure elucidation. In structure elucidation, you have to identify the structure of that compound for you to further modify it as well. However, this is quite expensive kasi the machine, the equipment being used is in the first place expensive. Okay? Ano yung mga pine-perform nating mga experiments to be able to identify the final structure of your lead compound? So there are two. The more common one is NMR spectroscopy. So later I will explain why. First, we have X-ray crystallography, and then second, we have NMR spectroscopy. So in the Philippines, mas common na ginagamit ang NMR spectroscopy because it's flexible or versatile sa compound na pwedeng i-test. Because in X-ray crystallography, it's only limited to the sample compounds na crystalline in structure. So what if your sample is oil-based? What if liquid-based siya? So medyo conflict na siya sa X-ray crystallography. Unlike NMR, it's again flexible with the type of sample that you have. It can be solid, it can be oil, or it can be liquid. That's why common ang NMR spectroscopy. Pero like what I said, ano na siya, tedious na talaga siya and it requires so much funds. Okay? So overall, that's the end of the first step in drug discovery. So ang first step natin, kung babalikan natin ang inyong mother outline, ito yung mother outline natin, the first step is finding the lead. So let's briefly recall the steps involved in finding the lead. Number one, what must be the priority in finding the lead? Do you need to find the lead outright? Yes or no? No, this is first. Okay, this is first. Very good. And then after identify the disease, you can extract, you can think of the possible T. Ano yung T? Targets. Very good, targets. And then after you identify the target, tetest mo ba agad sa tao? Yes or no? No. You need to perform first in vitro or in vivo. Vitro, very good, in vitro. Okay? And then, out of the test compounds that you had, once there are positive results, those compounds are said to be leads. Ayun na yung mga leads. And sa mga lead compounds na itinetest mo, meron kang iba't ibang ways to identify. So na-discuss na natin yon. And then purification if mixture pa yung inyong compound. And then identify the structure. So in that case, tapos na tayo sa finding the lead as the first general step. So proceed tayo sa second general step, that is drug design. So sa drug design, your raw material already is the lead. Meron ka ng lead. Pero anong sabi natin kanina? I repeat, lead compounds are not guaranteed 100% desirable. There are characteristics, there are properties of the lead compound that need to be modified, that need to be improved. So dyan papasok ang designing the drug. Okay, designing the drug. So ano yung mga considerations natin in drug design? So you have the lead compound, given nang active siya. So ano pa yung pwedeng gawin to make it better? Number one is to identify the so-called SAR, or the structure-activity relationship, followed by identifying the pharmacophore. Next is to improve its profile, or tawag natin doon drug optimization. Like for instance, improving dynamic and kinetic profiles. So let's start with the structure-activity relationship. What do we mean by that? Structure-activity relationship is defined as the relationship of how structural features of the molecule contribute to or take away from the desired biological activity. So the aim is to identify the parts of the molecule that are active and not active. Okay? So in SAR, aalamin natin kung ano yung part ng structure ng drug na active at hindi. So if medicinal chemists were able to identify the functional groups that are active, retained. Ay retained yon. Those that are observed to be undesirable, modify or remove if needed kung kinakailangang alisin. Those that are inactive, modify. Baka pag minodify, maging active. Okay? Yun yung identification of structure-activity relationship. Take note that SAR studies are essential in drug optimization because we can make activity or yung power ng gamot and selectivity of the drug better. Okay? 'Di ba? Goal natin ito in designing a drug to make it powerful and selective as much as possible. Okay? That's why we need to identify the structure-activity relationship. Identify the structures that are active and identify the structures that are inactive or toxic, let's say. If toxic, modify. If inactive, modify to make the drug better. Okay? That's SAR. Next, after you identify structure-activity relationship, you can now extract the so-called pharmacophore. Okay, pharmacophore. Ano ngayon ang pharmacophore? Pharmacophore, these are the binding groups that are active. Okay, these are the binding groups that are active. Important ba ang binding groups for a drug, for a compound to be active? Important ba siya? Yes. Ano bang meaning ng binding group? Ito ba yung nag-i-interact sa target? Ha? Yes, very good. So in pharmacophore, these are the binding groups or ito yung mga functional groups that are required for activity. So after you identify the functional groups that are active here o kaya hindi active, that's the time that you can get the pharmacophore. So whenever I say pharmacophore later on, ano na natin to, jargon na natin to sa subject natin, kapag mababanggit ko ang pharmacophore, mortal sin din sa inyo pag hindi nyo siya alam. Kasi pag sinabing pharmacophore, ito yung group, functional group na pumupunta sa target ng gamot. Okay? Pag sinabing pupunta sa target, yung active. Okay? Yung active. So that's identification of the pharmacophore. Okay? Kaya important din siya. So pag na-identify na ang pharmacophore, hindi dapat siya alisin. Pwede siyang i-modify lang to make it better din. Okay? To make it better. And then finally, once you identify pharmacophore and also the SAR, ay yung structure-activity relationship ng drug, you can optimize that already. Now, general lang yung nilagay ko dito. Examples of optimizations are dynamic, yung target optimization, that is to enhance the intermolecular bonds between the target and also the drug to be able to make the drug powerful and less toxic. And then, or mas maganda kung non-toxic, and then improve the ADME profile of the drug to ensure that the drug will reach its target. Okay? So marami siyang specific considerations pero I'll just leave its discussion sa inyong drug discovery na subject. May drug discovery kayong subject eh. Third year yon. Okay, third year. So at least ngayon meron kayong idea. Okay? So tapos na tayo sa drug design. Okay, drug design. So pag natapos natin ang drug design, confident ka na yung gamot ay uh, active. Okay? Confident ka na active siya at na-improve mo na siya. Ngayon, ang challenge mo na lang dito, paano mo siya mapapatunayan na active din sa iba't ibang uri ng tao sa buong mundo. Okay? Kumbaga, palaki na ng palaki ang scale. Of course, it could vary from one person to another kasi physiologically speaking, we differ. Like yung mga races, di ba? Yung mga blacks, different sa whites. Okay? Malaking factor yon. Kaya papasok dyan ang last general step called drug development. Now, in drug development, this is no longer my concern, to be honest, because may subject nga kayong drug discovery and development sa buong semester sa susunod na SEM, okay, sa third year life nyo. Kaya I'll just give you overview here, okay, some insights. Sa dosage form siguro na encounter nyo na, dito na tayo sa last slide, yung mga stages of drug development. So ang important lang sa akin dito is for you to identify the different stages. Stage 1, Stage 2, Stage 3, and Stage 4. Wherein in Stage 3, meron tayong iba't ibang phases. So paki-review nyo yung iba't ibang phases ng clinical trials. Yun yung 1, 2, 3, and 4. Okay, meron tayong phases 1, 2, 3, and 4. So I'll just leave this to you guys. I-search nyo lang kung ano yung mga names ng mga phases ng clinical trials. So you will be able to use that sa tasks, okay, na gagawin nyo sa Module 3. Okay? Yun lang, yung names lang nung phases. I-research nyo yan. Magagamit nyo yan sa task. Kung wala man sa task, sa exam, ay lalabas yon. Madali lang yan. Sa dosage form, meron din mga names yan. Okay? Or sa internet, meron. Ay napakadali. Yung mga specifics per stage, again, it's no longer my concern. Kasi sa medicinal chemistry, ang core ng discussion natin, in the interest of time, yung lead compound and yung designing the drug, lalo na yung SAR at yung pharmacophore. Okay? Kasi chemistry yun eh. Chemistry. Dito, hindi ko na masyadong tututukan. Okay? So overall, that's the end of Module 3, Drug Discovery and Design. To summarize our lecture on this module, so we have the current practice in drug discovery, design, and development. First is to find the lead. Second is to design the drug. Pag design the drug, pasok na yung chemistry ng gamot. Chemistry na talaga siya. Those are identification of SAR, and then pharmacophore and optimization, and then drug development. Okay? Taken all together, that's the end of Module 3. Okay? Tapos na yung drug discovery and design. Any questions? Naintindihan nyo guys? It's quite simple naman, no? Okay, if you will check. Okay na, goods tayo sa Module 3.